objection, the chair is authorized to declare recess at any time. Before I deliver my opening remarks, I wanted to note the unusual circumstances under which we're meeting today. Pursuant to Resolution 965, today the subcommittee will be meeting virtually. This is not how any of us would prefer to perform our duties, but remote work is unfortunately a necessity of the current moment and a reflection of the part we all have to play in slowing the spread of COVID-19. In light of this remote format, I want to offer some reminders to the members about the conduct of the hearing. Members should keep their video feed on as long as they are present at the hearing. Members are responsible for muting and unmuting their own microphones, and please keep your microphones muted unless you're speaking. You know, much as we love your family dog, there are limits here. And finally, if members have documents they wish to submit for the record, please email them to the committee clerk, whose email address was circulated prior to the hearing. Well, good afternoon and welcome to the first virtual hearing of the Committee on Investigations and Oversight. Today we're discussing a critical issue, research into repurposing of existing therapeutic drugs for COVID-19 treatment, as well as the scientific basis for the federal government's evaluation of such drugs. I appreciate our witnesses being here under these unusual circumstances, but these are very important issues and we look forward to your testimony. Today's hearing revolves around one of humanity's most promising tools in its public health response to the current pandemic, repurposing existing therapeutic drugs to treat COVID-19. The appeal of repurposing existing therapeutics is obvious. These drugs have already been developed, they've already been manufactured, and in some cases can quickly be accessed in large quantities. And for drugs that have already been approved to treat other diseases, a certain amount of safety data is often available to regulators. In the absence of any COVID-19 vaccine or novel treatment, existing therapeutics could potentially offer critical assistance for severely ill patients and bridge the gap until more prevention and treatment options become available. But with great promise comes great concerns. Since existing therapeutics rest at our fingertips and have demonstrated benefits in other circumstances, it can be all too easy in the midst of a pandemic to cut corners and to seek shortcuts to long-standing regulatory processes. And we can't allow that to happen. The evaluation process to repurpose approved drugs is there for a reason, to ensure that existing therapeutics, which could carry significant health risks for COVID-19 patients, are assessed through the prism of scientific and medical data and sanctioned on the basis of factual evidence regarding safety and efficacy in their new context. And while the process itself should be flexible and as fast as possible, the integrity of the process must be firmly upheld. The research community's evidence-based evaluation of existing therapeutics must be paramount, and political considerations must never enter into the equation for any specific treatment. Politics is allowed to interfere, the scientific research may be distorted, patients may be placed at risk, and the faith of the public in our whole public health mechanism may be shaken. Unfortunately, we're seeing the consequences of some political interference in the controversy surrounding two existing therapeutic drugs, chloroquine and hydroxychloroquine. In March, the FDA issued an emergency use authorization for these drugs as COVID-19 treatments. The scientific evidence to support this decision was dangerously thin, but the political considerations were clear, and our president became the world's loudest cheerleader for both drugs. Researchers, experts, and former FDA officials all question the decision for lacking a sufficient scientific basis. And now, nearly 3 months later, the FDA just this week revoked the emergency use authorization, acknowledging that clinical data showing the drugs, quote, may not be effective to treat COVID-19, and that, quote, the potential benefits of such use do not outweigh the known and potential risks. This is a clear example of the dangers of allowing political considerations to distort what should be a scientific process relying upon unbiased scientific evaluation. This hearing will explore the importance of supporting scientific research into repurposing existing therapeutics as COVID-19 treatments and the cost of neglecting science when politics intrudes. The research community is currently engaged in a heroic effort to explore as many therapeutics as possible in a search for COVID treatment. The federal government supports many of these efforts, but there may be more that we can do as policymakers to provide researchers with the funding and the conditions that they need to make progress. There may also be more that we can do to uphold the integrity and the role of science as the foundation for federal efforts in this area. Our witnesses bring diverse perspectives with deep experience in these areas. I look forward to learning from them about the most effective way for the federal government to support research into repurposing existing therapeutics and for this pandemic and unfortunately, probably for the next one. Well, thank you all. And the chair will now recognize Mr. Lucas, the chair of the full committee, for an opening statement.
Thank you, Chairman Foster, and thank you to our witnesses for their participation today. The COVID-19 pandemic is unlike anything we've faced since the 1918 Spanish flu. In those days, we had very few tools to slow the spread of the virus, develop treatments, or produce a vaccine to make ourselves immune to it. Thankfully, that has changed. Our nation's research enterprise, including government, academia, and industry, has the expertise, resources, and talent needed to fight this pandemic. The work they're doing to model the virus, screen potential treatments, and engineer new medical equipment is truly life-saving. We have supercomputers, advanced manufacturing techniques, and even advanced photon sources being used to fight COVID-19. From PPE, manufacturing, and new vaccine developments to repurposing existing therapeutics, America's scientific community has heeded the call to action. An excellent example of the public-private collaboration, leveraging technology to fight a common cause, is the COVID-19 High Performance Computing Consortium. Though this OSTP-led collaboration, COVID-19 researchers can access the world's most powerful computing resources to run complex models and develop large numbers of calculations at astonishing speeds. By leveraging these computing resources and deploying artificial intelligence and machine learning techniques, researchers can develop and determine, which drugs have the potential to be repurposed against COVID-19 at a speed and scale previously unthinkable. Technology will continue to play a critical role in saving lives and preventing the spread of COVID-19. And our federal research enterprise must have access to the resources and the technology necessary to do their jobs and to do it well. That's why I introduced the COVID Research Act of 2020, which would create an interagency working group and establish a national strategy to address infectious diseases. Additionally, this bill authorizes $50 million for DOE's infectious disease research program over the next 2 years. Working together with NASA and the NSF, this program gives us the ability to utilize the federal government's computing resources to respond to infectious diseases. Our national labs have already demonstrated the value of using high-performance computing and advanced research facilities to model the novel coronavirus, understand its effects on human cells, and predict its spread. I'm pleased to learn that there is work underway at Argonne National Labs that is particularly relevant to repurposing therapeutics to fight COVID-19. And thank you, Dr. Stevens, for being here today. I look forward to learning more about this important work. And more broadly, I'd like to extend my thanks to the entire scientific community, researcher after researcher, lab after lab, pivoting immediately to fight COVID-19 when it reached our shores. When I began serving as ranking member of the committee, I said that one of our most important responsibilities is to tell the story of science and to make sure our constituents understand the tremendous research being done and why it matters to the next generation of Americans. This story in particular, how American scientists, researchers, and engineers responded to COVID-19, is one everyone should know. And I hope my colleagues will use this hearing as one more opportunity to share this work. And with that, Mr. Chairman, I yield back.
Thank you. And if there are members who wish to submit additional opening statements, your statements will be added to the record at this point. At this point, I'd like to introduce the witnesses. Our first witness is Dr. Peter Lurie. Dr. Laurie is the president of the Center for Science and the Public Interest, a nonprofit health advocacy group based in Washington, D.C. Before joining CSPI, he held several positions at the Food and Drug Administration, including a stint as the FDA's Associate Commissioner for Public Health Strategy and Analysis. He served nearly 8 years as a top official of the FDA. Our next witness is Dr. James Finnegan. Dr. Finnegan is the director of the Respiratory Centers of Excellence at National Jewish Health, the nation's leading respiratory hospital. He's also the medical director of the lung cancer screening program at the National Jewish Health. Dr. Finnegan is a pulmonologist with a research focus on lung cancer and injury. Our third witness is Dr. Rick Stevens. Dr. Stevens is the Associate Laboratory Director for Computing, Environment, and Life Sciences at Argonne National Laboratory. He also serves as the leader of Argonne's Exascale Computing Initiative. He's worked at Argonne Lab since 1982. Our final witness is Dr. Benjamin Rome. Dr. Rome is an associate physician at Brigham and Williams Women's Hospital in Boston, Massachusetts. He's also a postdoctoral research fellow at Harvard Medical School. Dr. Rome's academic research focuses on the FDA approval process for drugs and medical devices, as well as the effect of federal policies and regulations on drug pricing and utilization. As our witnesses should know, each of you have 5 minutes for your spoken testimony. Your written testimony will be included in the record for the hearing. And when you've all completed your spoken testimony, we'll begin with questions. Each member will have 5 minutes to question the panel. And if there is time, we may be able to have a second round of questions from those members who are in attendance. And we'll now start with Dr. Laurie.
Well, thank you, Chairman Foster, Ranking Member Lucas, and other committee members for inviting me to testify on this important topic. For this testimony, I'm defining repurposed drugs as approved drugs for which a second indication for COVID-19 is sought. Some other witnesses may use other definitions. Unfortunately, the unmistakable allure of repurposed drugs is not enough on its own. Effectiveness for one condition does not guarantee effectiveness for a second, even closely related condition. Target populations may be demographically and medically different, and so even the existing safety bases may have only limited relevance. The product may be administered in different doses or by different routes than for the first condition. So let's look at 2 repurposed drugs. The antimalarial drugs, chloroquine and hydroxychloroquine, which I'm considering together, would likely have languished well down the list of candidates for COVID-19 had they not been catapulted to prominence by President Trump's comments. On March 21st, the president described them as, quote, one of the biggest game changers in the history of medicine, and later stated that he was taking the drug himself, the ultimate celebrity endorsement. On March 28th, under pressure from the administration, FDA granted the drugs an emergency use authorization, EUA, but the evidence provided for this EUA was less than that provided for many previous EUAs. Eventually, the scientific process played itself out. There were several observational studies that demonstrated either no benefit for the drugs or even indicated that mortality rates were higher. FDA issued a warning that the drugs caused life-threatening arrhythmias. And finally, there were 2 randomized controlled trials, the gold standard for scientific evidence. The first suggested that the product was ineffective in preventing infection among those exposed to the virus, and the second that it was also ineffective in treating SARS-CoV-2 infection itself. On June 15th, FDA revoked the EUA. What can we learn from this embarrassing? Well, first, we should adhere to accepted methods of drug discovery, even in a pandemic. It is the painstaking process of conducting randomized controlled trials that ultimately produces definitive evidence, even if it is definitive evidence of lack of effectiveness. Second, that patients, in the president's phrase, did have a lot to lose. Life-threatening arrhythmias were fairly common. Even patients without COVID-19 suffered, as those needing hydroxychloroquine for its FDA-approved conditions had difficulty obtaining the drug due to increased demand. Finally, the president's announcements distorted the overall research effort for COVID-19. It is inconceivable that left to their own devices, scientists would have designed over 150 randomized controlled trials assessing the effectiveness of these drugs. How many more promising drugs were left unstudied or understudied as researchers pivoted to address the headlines? A second problematic repurposed drug is famotidine, an over-the-counter heartburn drug, also known as Pepcid, a seemingly unlikely drug for COVID-19. One of its primary advocates is a Boston physician named Dr. Michael Callahan, who is also a consultant on the staff of the Assistant Secretary for Preparedness and Response, ASPR, Dr. Robert Kadlec. Under Dr. Kadlec's direction, Dr. Callahan, having advocated for the drug, assisted a pharmaceutical company and a hospital to prepare an application for funding to ASPR to conduct a trial of famotidine. Shortly thereafter, Dr. Kadlec ordered a hefty $21 million contract to these entities. Senior officials were cut out of the granting process. There are two other prominent drugs worth mentioning here. The first, remdesivir, is the only drug so far proved effective against SARS-CoV-2, but it is not a repurposed drug, at least in my definition. Rather, it is an unapproved drug with known antiviral activity demonstrated to be effective in a study funded by NIH. The second drug, dexamethasone, is the first drug to reduce mortality in patients with COVID-19. Whether it's a repurposed drug is a matter of definition, as it's long been approved, but is often considered as a general treatment for severe respiratory illness based on its anti-inflammatory activity and is not directed at SARS-CoV-2 itself. But the benefits of these two drugs were demonstrated the old-fashioned way, through rigorous randomized controlled trials. Interestingly, the dexamethasone results are derived from the same British study that reported the ineffectiveness of hydroxychloroquine. That trial is very large and able to test multiple candidate therapies simultaneously. In contrast, the clinical trials effort in the United States has been fragmented and poorly prioritized, resulting in many relatively small studies, often testing the same drugs, with some studies struggling to enroll patients. In conclusion, so far in this pandemic, effective treatments have not been identified by anecdote, by wishful thinking, by presidential pronouncement, or by questionable contracting practices. They were identified instead by the fair, transparent, and systematic application of the very scientific principles that for decades have delivered so many safe and effective treatments. But when we departed from these principles, precious time was lost, resources were squandered, and some patients paid with their lives. Thank you.
Thank you. And our next witness is Dr. Finnegan. Now recognized for 5 minutes.
Thank you. I would like to thank the members of the subcommittee for inviting me to speak on my experiences as a clinical investigator during the COVID-19 pandemic. I am a pulmonary and critical care physician at National Jewish Health in Denver, Colorado, where I see patients in our pulmonary clinic and intensive care units. National Jewish Health is the leading respiratory hospital in the nation and is the only facility in the world dedicated exclusively to medical research and to the treatment of patients with respiratory, cardiac, immune, and related disorders. We work with several hospitals in Colorado, including our flagship St. Joseph Hospital, to provide pulmonary and critical care medicine, and have established respiratory institutes in New York in partnership with Mount Sinai Health System and in Philadelphia with Jefferson Health. I have close to 2 decades of basic, translational, and clinical research experience and currently help lead our COVID-19 clinical research program. Responding to this pandemic has required a complete reorientation of our clinical and research programs at National Jewish. Clinically, we reorganized our workforce and physical plant to diagnose and treat COVID-19 patients while simultaneously planning for the worst-case scenario. For our research operations, it meant halting existing studies and starting up new studies as quickly as possible, all with much of our staff working remotely. Many of these studies are basic science investigations to identify new targets for treatment. At National Jewish Health, we've gone from 0 COVID-19 clinical studies to 10 or more therapeutic trials in various stages of development over the past 12 weeks. Prior to embarking on any specific research study, each trial requires a number of time-consuming steps, including protocol review, assessment of our ability to perform the trial as designed, determination of any conflicts with ongoing trials, budget negotiation, and agreement on contracting terms. This process ordinarily takes 3 months or longer. During this crisis, we've been able to cut that time to a few weeks. For a pharmaceutical company or other study sponsor, this process must be repeated at every study site. As an example, the recently published remdesivir study had 60 sites. In reflecting on our experience at National Jewish Health during this pandemic, I believe 3 points should be highlighted. First, to rapidly deploy clinical trials of new or repurposed drugs, a pre-existing organized network of research sites is essential. The recently announced NIH-led ACTIVE program is an example of this. ACTIVE stands for Accelerating COVID-19 Therapeutic Interventions and Vaccines and is a public-private partnership to create a collaborative framework for prioritizing and streamlining clinical trials using existing clinical networks. Another example is the Prevention and Treatment of Acute Lung Injury, or PEDAL network, an NIH-funded network of academic medical centers dedicated to studying acute lung injury and the acute respiratory distress syndrome, the disease caused by SARS-CoV-2. This network has been repurposed to study the clinical features and possible treatments of COVID-19. For the past month, the PEDAL network developed and launched 2 research protocols, and both studies will likely be completed in the coming weeks. Networks like these can be used in collaboration with industry as a platform to launch quickly new studies on promising treatments. Second, we need ongoing investigation of SARS-CoV-2 and COVID-19 to understand the virus and mechanisms of this disease. Much of this research will be what we call preclinical studies, bench research in cells and animal models to expand our understanding of COVID-19 pathophysiology. However, this can only exist if we maintain a robust national medical research mission and infrastructure. Dividends from this kind of research are not always immediately apparent. However, a basic understanding of the underlying science of this disease will drive development of new therapeutics moving forward, both for this pandemic and to prepare us for the next one. These studies can help identify which new drugs are most promising and can inform a rational strategy for prioritizing drugs for clinical trials. Third, another pandemic is likely in our future. What that will be, we don't know, but we should be planning now on how to incorporate a full research operation into any future pandemic response. I've been impressed with the research community reaction to this crisis. However, even with this effort, an organized national response was not launched until several months into the pandemic. Coordination of what research will be performed and how it will be executed, the respective roles of organizations such as the FDA, NIH, CDC, BARDA, as well as industry should be considered prospectively. Research is as important to defeating this pandemic and being ready for the next one as personal protective equipment, intensive care units, and ventilators. Thank you.
Thank you. That was beautifully timed. The chairman appreciates the accuracy of your time estimate, and we'll now recognize Dr. Stevens for 5 minutes. Whoops. Rick, mute.
Okay, I thought they were going to unmute me. So thank you, Chairman Foster and Ranking Member Lucas and members of the subcommittee for inviting me here to talk about our work related to COVID-19. My group and collaborators work primarily in technical components of this, so let me talk a little bit about what we're doing. I should say that I'm speaking for Argonne, myself and for Argonne. I'm not speaking for the Department of Energy. My work focuses on applying high-performance computing methods to problems in science and medicine, and I've worked in this combination space for over 25 years. And related to COVID-19, I'm the co-PI on a DOE CARES Act-funded 9-laboratory consortium project that is working on molecular design for COVID. As part of that effort, we're looking at repurposable drugs as well as de novo compounds. And let me tell you a little bit about the virus and why this drug search process is actually challenging. The virus is a single-stranded RNA virus. It codes for about 30 proteins. About 2/3 of those proteins commandeer host cell machinery to make copies of the virus, and about 1/3 of them are involved in packaging and formulating the virus. Of those proteins, there's perhaps 10, maybe a little bit more than that, in the virus that the virus codes for that are plausible drug targets. The virus proteins also interact with the host. Perhaps as many as 300 protein interactions appear to occur, and a number of those host proteins could also be drug targets. Now, it's also important to know that this virus is very closely related to SARS-1, and so since around 2003, the community has had access to information about its genome and its proteome and has been working on this. And thanks to the DOE light sources and light sources elsewhere, we have very detailed atomic structural maps of these proteins. And in the last few months, we've acquired more of these structural maps. But what's not known is essentially how existing drugs interact with these virus targets. That has not been the subject of large-scale computational work prior to now, and has not been the study of large-scale experimental work. So what I'm working in, and my collaborators at the University of Chicago and at the 9 national laboratories that are collaborating with us, are really looking at how we can apply high-performance computing to scan not only the 2,500 or so licensed drugs worldwide and the 7,000 or so drugs that are in the pipeline, but literally billions of molecules that we know can exist. Now, in this effort, it's also critical to recognize the 3 main infrastructures that we're using. We're using supercomputers, the fastest machines in the world that exist at the DOE labs, like at Argonne and at Oak Ridge. The famous Summit computer at Oak Ridge is a workhorse for us, but also machines that the NSF supports. We also use the light sources. These are critical for determining structures of proteins and for determining structure with molecules, potentially inhibitors bound into these proteins, so we can understand their mechanism of action. And the third resource that's critical for this effort is the NIH-funded biocontainment laboratories, the regional biocontainment laboratories that were stood up after 9/11, after the scare, after the anthrax scare, and the emerging concern about emerging pathogens. And these exist at various locations in the country. The one that is close to me is the H.T. Ricketts Lab, operated by the University of Chicago here at Argonne, but we're also using the Regional Biocontainment Laboratory that's at the University of Tennessee Health Sciences Center. Those centers are critical because they can work on active virus that's essentially, I mean, viruses are not alive, but viruses where we can look at the entire life cycle in whole cell assays and animal-based assays. So essentially, what our program is to use the computers to search for molecules, including all the repurposing drugs. They get scored on each individual molecular target. The drugs that appear to have high potential get forwarded to our experimental collaborators, where they get assayed biochemically and then ultimately through whole cell assays. I should point out one more thing. There's lots of advice we could give about future pandemics, but one thing that's holding us back, that's holding back the scientific community right now, is the lack of biochemical assays for these specific virus protein functions. The National Institutes of Health are investing via the NCATS program in that. The national laboratories are investing in that, but that is a major bottleneck. In future, we need to invest in assays. They need to be stockpiled. They should become part of the national critical infrastructure and made available to these biocontainment laboratories so that in the future, rapid screening can happen on any new outbreak. And I'll leave it at that. Thank you.
Well, thank you. Well, at this point, we'll begin our first round of questions, and the chair will recognize himself for 5 minutes here. Dr. Rome and Dr. Lurie, there's been mention of what is being set up in Europe and internationally, and as opposed to what has happened in the United States in terms of the coordination of the large number of clinical trials. Could either of you say a little bit about what we get right that's gotten that's better or worse than what's done in other countries and other international collaborations, and whether we're insufficiently or sufficiently connected to those?
Let me let Mr. Dr. Rome answer, because, sir, I think you neglected to give him the chance to read his statement.
Oh, goodness.
Mr. Chairman, you didn't.
My apologies. Thank you, Dr. Rome. I was fumbling multiple windows here and neglected. So, and thank you for that, Dr. Laurie. All right, Dr. Rome, you're now recognized for 5 minutes.
Thank you. Sorry about that. So, Chairman Foster, Ranking Member Lucas, and members of the subcommittee, thank you for inviting me. I am a practicing primary care physician and a health policy researcher at Harvard Medical School and Brigham and Women's Hospital in Boston. I am a member of the Division of Pharmacoepidemiology and Pharmacoeconomics and the Program on Regulation, Therapeutics, and Law, or PORTAL, an interdisciplinary research group that studies prescription drug development, regulation, use, and cost. I'm honored to be here today to talk with you about the process for studying and approving repurposed drugs during the COVID-19 pandemic. Drug development can be a lengthy process, and repurposing several medications with existing data about safe use in humans allowed clinical trials to begin early in the pandemic. As a result, just 4 months after the first COVID-19 patient was reported in the US, several high-quality clinical trials have provided solid evidence relating to at least 4 drugs, 2 of which have proven effective: dexamethasone, a low-cost generic corticosteroid that can be readily prescribed by clinicians, and remdesivir, an antiviral that has not yet been approved for any indication by the FDA, but is now available under an emergency use authorization. However, we have also witnessed examples of how the process for testing and approving drugs can go awry, as exemplified by the case of hydroxychloroquine. We should learn from our past missteps as we move forward, and our experiences so far suggest 4 key actions Congress should take. First, Congress should hold all government agencies and officials accountable for making statements and acting based on the best available scientific evidence. Hydroxychloroquine was widely touted by President Donald Trump and was issued an emergency use authorization by the FDA based on preclinical and limited anecdotal evidence that turned out to be unreliable. These actions led to widespread use of the drug, which exposed patients to risk, led to shortages at pharmacies, and diverted attention and resources that might have been dedicated to other potential therapies. Second, Congress should invest heavily in high-quality clinical trials, which are necessary for determining whether drugs are safe and effective. Notably, most of the high-quality evidence generated so far during the pandemic has resulted from public funding, including the US government in the case of remdesivir and the United Kingdom government in the case of dexamethasone. While the pharmaceutical industry will continue to have a role to play, the federal government's leadership and involvement are crucial, particularly for repurposed drugs, which industry may have little or no financial incentive to study. However, such public investment should be made with the assurance that any medications that are found effective will be priced fairly and distributed equitably to patients who need them. No American should be prevented from accessing potentially life-saving treatment for COVID-19 due to cost. Third, Congress should invest in a public health infrastructure and national clinical trial network that can help shape the research agenda, facilitate research across multiple sites, and limit duplicative efforts. In several European countries, government and academics have collaborated on large clinical trials that test multiple repurposed drugs simultaneously. A prime example is the Recovery trial, based out of the University of Oxford. This trial has already provided useful information about the lack of effectiveness of hydroxychloroquine and the effectiveness of dexamethasone. Finally, Congress should amend the process by which the FDA issued emergency use authorizations, or EUAs. The level of evidence required to meet the standard of an EUA should be clarified. And to increase transparency, Congress should compel the FDA to make all related data public at the time an EUA is issued. As new evidence emerges, the FDA should be directed to apply the same standards for revoking an EUA as was required for issuing it. EUA should be accompanied by a clear and transparent plan for how the drug will be fairly and equitably distributed to patients, something that was lacking for both hydroxychloroquine and remdesivir. Finally, issuance of an EUA should be accompanied by collection of data on treated patients to gain additional insight about the drug's safety and effectiveness. Our experiences so far studying repurposed drugs during the COVID-19 pandemic have shown that we need not choose between rigorous scientific evidence and speed. We can have both. As our fight to control the COVID-19 pandemic continues, Congress must assure that we uphold a drug approval process that follows the science and promotes the practice of evidence-based medicine. In a recent viewpoint published in the New England Journal of Medicine, Dr. Jerry Aborn and I argued that, quote, the health of individual patients and the public at large will be best served by remaining true to our time-tested approach to clinical trial evidence and drug evaluation. Thank you.
Thank you. And my apologies again for having skipped your testimony in the order. That's I guess, the danger that occurs when I've read your testimony in advance and feel less need to hear it directly from you. And so I guess I will repeat my question, which was largely directed at your testimony, to say a little bit more about the differences in the way things are being done internationally versus in the U.S. and what lessons we might learn from that.
Thank you. Yeah, I'm appreciative. So your question is a good one. We have had two successes in this pandemic so far with dexamethasone and remdesivir. One was primarily driven out of the U.S., but the other, dexamethasone, came from funding from other countries. And I can say that most of the large clinical trials that are being conducted, especially the ones that were started very, very early in the pandemic and are likely to get us results in a timely fashion, were led by our peers in Europe and other countries. And definitely the U.S. should and going forward in this pandemic and in future pandemics, lead those efforts and become a leader in the world for running these sorts of clinical trials.
Now, in the case of the drug that worked, the anti-inflammatory that worked in England, was that funded by the manufacturer? Because one of the things that worries me here is that if the majority of our clinical trials are funded by manufacturers, you will naturally, they will prefer drugs for which they have an intellectual property or a manufacturing position in. And that we may forego equally promising drugs that are, for example, off patent or just become generic drugs. And is that handled differently in other countries in the U.S., or are essentially all trials worldwide funded by the manufacturer?
No, not all trials are funded by the manufacturer, and the trial out of Oxford was funded by the UK government. Dexamethasone is a generic drug. There's very little skin in the game for any manufacturer to conduct a clinical trial. And that's likely to be true of many of these repurposed drugs, which may be older, off-patent, generic drugs, which are widely available and great for patients, but not necessarily a good investment from the perspective of a manufacturer. But not only that, the clinical trial for remdesivir, which is still a patented drug, owned by Gilead, the key clinical trial upon which we are using that drug and the EUA was based was funded by the U.S. government, by the NIAID. And this is a case where Gilead should have been running the key clinical trials and instead ran a clinical trial testing two different doses of the medication against itself, leaving the key clinical trial to come to the U.S. government. And they have a patent on that drug and can financially benefit from it when or if and when it gets FDA approved.
Thank you. Yeah, Dr. Lurie, any comments on what's done internationally versus the U.S. and lessons we might learn?
Well, I think what Dr. Rome said is spot on the money. Unfortunately, things have been, to a certain extent, delegated to the pharmaceutical industry and even to individual academic institutions in this country. What we really need is somebody to coordinate the whole thing. I know that the NIH would like to do that. I know that the British government has done so very successfully, particularly in the recovery trial, but we need a stronger hand on the rudder. Somebody to prioritize the drugs, figure out the ones that matter the most, and then do proper trials on them. I guess I'd add to what Dr. Rome said in the following way. It's tempting in the setting of a pandemic to say that we need to cut corners. But actually, the pandemic is almost the best place to stick with the usual game plan because there are so many patients, I'm sorry to say, and the disease makes you ill so quickly, I'm sorry to say, that there's more than enough statistical power for people to do the randomized controlled trials, which we expect drug approval to be based upon. So let's stick with the methods that work. Let's not use the pandemic as some excuse for corner cutting or for deregulation.
All right. So, yeah, that's interesting. Do you think that there may be opportunities that we're overlooking for sort of lightweight outpatient clinical trials? You know, you can imagine to have a quick but scientifically valid look at hydroxychloroquine, for example. If you had some fraction of the large number of people who tested positive were immediately given the option of being screened and just on an outpatient basis, given that we could have rapidly understood and, you know, without a lot of safety concerns. Are there opportunities there we should think about?
I think there are, but so far, if you look at the totality of the research that's been done, most of it is based in the inpatient setting. And that makes a certain kind of sense. These are the sickest patients, after all. They're the patients in whom you can demonstrate benefit most easily. So I agree there's opportunity, but I don't mean that as a criticism of the research enterprise to date.
All right, and that exceeds my 5 minutes, and so I'd like to recognize Representative Lucas, Chairman Lucas, for 5 minutes.
Thank you, Mr. Chairman. Dr. Stevens, could you provide us with an example of cutting-edge research capability unique to the Argonne National Lab and how it's helping researchers work forward towards repurposing existing drugs to treat COVID-19?
Well, probably the best example is the use of the advanced photon source to quickly, within a few days, assuming we can get a good crystal, produce a new structure of a viral protein bound to a drug. And this is critical to understand whether or not the target is in fact the target that the drug is working on, to understand how it changes the structure of the protein, how it might interrupt its function. So that's a pretty unique capability. There's only a handful of places in the US and in the world that can do that and can do it quickly. And after the pandemic became apparent, and Argonne went into a lowered level of operation, we kept the advanced photon source up, and we kept that team working on determining protein structure through the virus intact. So that's a really good example, I think.
Mm-hmm. Well, could you, along those lines, explain how Argonne National Lab is working collaboratively with other research entities towards identifying COVID-19 drug candidates?
So we have a 9-lab consortium that includes Argonne, Oak Ridge, Berkeley, Brookhaven, Los Alamos, Livermore, Sandia National Laboratories, SLAC, and Pacific Northwest Labs. 9 labs all working together. We are using large-scale computation to computationally screen drugs, billions of drugs, or billions of potential drugs. All the existing drugs and billions of molecules that become potential drugs. The drugs that score hits, interesting from that computation, are immediately procured in some fashion or synthesized in some cases if you can't get them fast enough, and turned over to experimentalists that can assay the function of that inhibitor. And for things that succeed in that test, then get progressed to whole cell assays with full virus replication. Things that would succeed there would get passed on to animal models and then passed on to clinical activity. I should point out that it takes a large team to do this with virologists, physicians, medicinal chemists, computer scientists, and so on. And because of that large team, in fact, even the indications of interesting compounds or existing drugs were discussed with physicians that were actually on the clinical front line. And so we were benefiting from their advice, and then benefiting from early information we could bring into the clinical use if necessary.
As I mentioned earlier, my bill, H.R. 6599, focuses on collaboration among federal agencies and aims to create a national strategy for any infectious disease outbreaks we face in the future. And certainly we will face those in the future again, as the chairman alluded to. Are there any other collaborations that we're not thinking about that would help you, Dr. Stevens?
Well, early on, we established a collaboration between the DOE National Lab, the NIH, which has laboratory activities and research groups. So there's a linkage. I think what's needed in the framework of your bill is a sustained, ongoing network of research collaborations that's consistently working on problems and is sharing data, sharing reagents, sharing assays, sharing information. And it would involve the DOE, involve the NIH, it probably would involve NSF resources in some cases, the DoD, essentially all the players that are necessary for the integrated public health system in this country in some kind of ongoing collaboration around emerging threats.
One last question, Dr. Stevens. Is there anything Congress can do to help scientists and researchers like yourself do their jobs more efficiently during this pandemic and bring us one step closer to ultimately a cure?
I believe that the current activity is exactly what's needed. The funding, the ongoing funding support for these New efforts is, of course, critical to extend that longer. We're not out of this pandemic yet. I believe the interagency coordination process is working quite well. I believe that the institutions have put together structures to support this. Within the Department of Energy, for example, they created the National Virtual Biotechnology Lab coordination process across all the national labs in the U.S. to drive them towards a coherent COVID-19 strategy. So I believe that's working. So continued support for that level of activity is what I recommend.
Thank you, Doctor. Thank you, Mr. Chairman. Yield back.
Thank you. And I will now recognize Representative Bonamici for 5 minutes.
Thank you, Mr. Chairman. Can you hear me?
Yes.
Terrific. Well, thank you to Chair Foster and Ranking Member Lucas, and really to our witnesses today. Thank you for your expertise. We know that COVID-19 is disproportionately affecting Black, Latinx, Indigenous, and other people of color, and that they are contracting the disease and dying at alarming rates. And as researchers develop studies and carry out trials, the administration and Congress can and must address these disparities and do all we can to encourage or require research that's deliberately inclusive of all demographics and effectively addresses these inequitable outcomes. Recruiting patients to participate in research, we understand, can be challenging even under ideal settings, but trials can fail if there's a lack of patient enrollment. There are valid reasons why a patient invited to take place in a study may not trust medical researchers or feel comfortable participating. And even once a willing participant is identified, there can be barriers to their inclusion. A recent Politico article talked about physicians who are conducting NIH-funded clinical trials for remdesivir at sites in Boston, New York, and Atlanta. They faced language barriers in recruiting patients with limited English proficiency. They didn't have consent forms in Spanish. They had to work with translators by phone to explain the study and get consent, and it took extra hours per patient. So I know, Dr. Lurie, you mentioned this. Do the witnesses all agree that it's important to have diverse representation in studies and trials? Do you all acknowledge that that's important?
Well, if I may go, I most certainly do. I've not seen specific information about recruitment and how it might vary by ethnicity so far in this pandemic, but I do know that this is not an equal opportunity virus. At all stages of this disease, African Americans and other people of color are at a distinct disadvantage. They're more likely to be exposed to the virus in that they're more likely to be living in crowded living conditions, more likely to be working in meatpacking plants, more likely to be in prison, more likely to be healthcare workers at the front lines of exposure to the virus. After that, they'll very often have difficulty gaining access to treatment, in part because of lack of health insurance. And then within the hospital, the outcomes have not been equal across ethnic groups, perhaps because of significant underlying health conditions as well. So all the way across.
I appreciate that. I'm going to ask, I don't mean to interrupt Dr. Laurie, but I'm also going to ask you and Dr. Finnegan, are there strategies to get diverse representation in testing and trials? And if not, what could not only the government, but what can the medical community do? Do you have suggestions on how to build trust in the communities that may have been historically excluded from government-funded research? I want to get your thoughts on this with Dr. Finnegan.
Well, Dr. Finnegan, go ahead.
Okay, thank you. So first off, you're correct. So I've seen firsthand in the COVID-19 pandemic where there have been examples of people who were not recruited adequately or were not recruited because there was a language barrier, there was not an appropriate consent form that was particularly challenging for people who only spoke Spanish. One important thing to note is that there's always a time window in which you can recruit somebody. So typically you can't recruit somebody into a study in an unlimited time window. You may have to do it within, let's say, 24 or 48 hours of them being positive. And so there's a little bit of a race against the clock in this. And so those kinds of barriers really can have significant meaning. I think those things need to be taken account ahead of time. It is relatively common that African Americans, Latinos, non-whites are sort of underrepresented in studies. And so being ready for that kind of thing ahead of time to make sure they can get adequately enrolled, all the resources are available to them. Those things are critical. I know of studies where they got those kinds of resources, those consent forms, but it happened weeks into the trial. And so you lost time, you lost patients. And the other critical piece there that I think you're also getting at is that's a lost opportunity for that person to be in a trial. Our goal is to give every patient the opportunity to enroll in a trial if they want to. And if you can't enroll them because there's not an adequate consent form, let's say, that's a missed opportunity for that patient, and they may lose the chance of being in a trial, let's say on remdesivir, where there's a potential benefit.
I appreciate that. And there was just an article this morning, Axios Health newsletter, that there's all this talk this week about the steroid that's been hailed as a breakthrough treatment. But there's also evidence, according to the chair of of pharmacology at Johns Hopkins University School of Medicine, that African Americans may respond differently to this type of steroid. So it's so critical that we address this issue because, as we know, COVID-19 doesn't discriminate based on somebody's race. I mean, it's disproportionately affecting people of color. Real quickly, Dr. Finnegan, we know that repurposing existing drugs is an attractive option because medications have already gone through testing. Can you tell us more about how preclinical testing and clinical trial phases can be safely accelerated if the drug has already been approved for another use?
So there are different ways this can be done, but I think the key is to have sort of a integrated network so you can hit all the phases at once. So as has been described, there are things you can do in silico in a computer where you can try to rationally identify drugs. So for example, I was a part of a study where one of our investigators built essentially a Google program where you could put in drugs and then put in the kind of drug you wanted or the kind of protein you wanted to target. And then this computer program would sort of spit out the drug that you wanted. And then you could marry that to an assay that you could do in a lab where you might have, let's say, 500 little wells, and you could test different drugs in each one of those wells. And so being able to marry those different things together and being able to do that and then rapidly move that into an animal model, those things are important to have that whole spectrum represented. Whether or not that requires one institution or multiple institutions is important. And I think, as was said earlier, having these assays sort of ready to go is important. And so thinking about what these might be, they often have to do with toxicity, whether or not they kill cells or something like that. Those are the things that need to be thought about ahead of time so they can be ready to go.
So I'm afraid I have to.
My time is expired. I yield back, Mr. Chairman. Thank you.
That's right. And we'll have a little bit of forbearance with representatives who are coming in on the telephone because they can't see the timer. And we'll now recognize Representative Biggs for 5 minutes.
Thank you, Mr. Chairman, and thanks, Ranking Member Lucas, and thank you to each member of the panel. This has been very interesting. And as we're trying to develop vaccine or cures for the COVID, I see we're moving fairly rapidly. It looks like we're moving rapidly, and I appreciate Dr. Lurie talking about through this maintaining some standards and normalcy. But I guess my question that I want to ask here is as we go through this and we accelerate these processes, can you address, and I guess we'll go to, I'll just open it up to whoever wants to address, answer this. How can we maintain scientific rigor while we're accelerating the development of vaccines and curatives? And each one of you kind of have nibbled around the edges here, but I'm wondering if there's any way that you see that we can maintain that scientific rigor that's so necessary if we're going to really get a handle on this.
Well, I think the playbook is clear. The playbook is the playbook that we've had for decades, the playbook that has produced effective therapy through following that very playbook, through keeping to strong standards. So all I think we really need to do is to coordinate better, obviously speed things up the way researchers collectively have decided to do by turning their attention to this. And if we keep our standards up, I like to think that ultimately we'll have the products that we need.
Well, and so that gets me to Dr. Rome, who I think mentioned something I'm in total agreement with. If you'd maybe expand on that, you said in EUAs, one of the things that we need to do is be more transparent. And so, Dr. Rome, if you would just address that, how we do that, and how can we best be more transparent?
Yeah, thank you. I totally agree. Transparency is key with EUAs. We've had two examples of EUAs, or emergency use authorizations, so far during this pandemic. So hydroxychloroquine and chloroquine was the first one. That was based on very little evidence, but when the EUA came out, it wasn't clear what evidence the FDA had considered, and it was sort of only later did we sort of learn a little bit about what was going through the minds of the folks at the FDA. With remdesivir, we had a top-line result published by the clinical trial, but not until 3 weeks later did we have the full data that was released in the New England Journal of Medicine. And during those 3 weeks, the drug was already being shipped out to hospitals and used, and we didn't know necessarily how to best target the drug towards particular patients, or if there was, and we had a limited supply. So absolutely, transparency is key. The FDA should make decisions in a consistent way that meet the standard. The standard for an EUA is that it is reasonable to believe, based on the totality of evidence, that the drug is likely to be effective. So that reasonable to believe standard needs to be enforced. When new data comes out, we need to reassess, and we need the whatever data that the FDA is using to make those decisions should be public at the same moment that the EUA letter is issued.
Great. Thank you. And I'm just going to go to Dr. Stevens. Dr. Stevens, you mentioned that although repurposing existing drugs may be a fast route to possible treatments, it's highly likely that we're going to still need purpose-built drugs for treating COVID-19. Can you expand on how the work that you're doing at Argonne be leveraged by pharmaceutical companies to help develop purpose-built drugs specifically?
Yeah, so what the team is doing is we are trying to produce a set of qualified leads. That would be compounds that show some promise in the computational work, they show some promise in initial functional assays and initial wholesale assays, but are not fully refined as drugs. These would be compounds that we would hand over essentially to the pharmaceutical industry and say, here's all the data that we've computed and that we've measured on these compounds, and let them take it from there. And this that kind of handoff has been discussed between the labs and pharma, and it's the kind of thing that pharma is very interested in, because you think of the drug development process as a giant funnel, and at the top of the funnel, you've got billions of possible molecules. Out of 10 to the 60th possible drug molecules, we know of a few billion of them that we've thought about, in some sense. And we have to narrow that down to handfuls, tens or 20s of compounds that pharmaceutical can take and do more advanced studies on, can refine and improve the molecules, improve their safety, improve their effectiveness, improve the therapeutic window. And so what we're doing in the public sector is essentially that big top part of the funnel and reducing it down in a very public way, a very open way, a set of priorities that they can then take and invest private money into, refine to drugs. That's the strategy.
Great. Thank you. And with that, Mr. Chairman, I'll yield back.
Thank you. And I will now recognize Mr. Byer for 5 minutes.
Thank you, Mr. Chairman, and thanks all of you for being with us. Let me begin with Dr. Rome. One of the things in your testimony, you said it's imperative that we establish a clinical trial network. Does the one not already exist, and isn't that part of the function of NIH to have established that over the years?
Thank you for the question. Yeah, so certainly we have small networks of clinical trialists, and I think that Dr. Finnegan could probably speak to this more as a trialist himself. But I would say that the NIH does in fact do direct research itself in some cases, as was the case in remdesivir. But in other cases, it will outsource to academics. And as was already mentioned, when a study is done at multiple sites, there's a lot of regulatory things that need to happen. You need to assure safety of the clinical trial through institutional review boards, and that can take a long time. And so by what I think where I think the efforts need to be is clinical trialists and scientists are ready to go and want to act. What needs to be cut down on are the things that are super important, like maintaining patient safety. But the time to do those things and the time to coordinate across different sites can definitely be cut down on by investing further in those infrastructures.
The vision has to be that there is an existing national network of clinical sites, say 50, 100, 300, that in a future crisis click, you turn it on. So, Dr. Finnegan, you mentioned that at National Jewish Health, you're working on delineating the structure of the protein. Why would an individual hospital do that rather than the national labs that have all the big computing machines and the neutron devices and the light devices?
It has to do with a couple things. Number one, ability and interest. So we have people who have that ability and can do it. And as I said, everything sort of reoriented towards COVID-19. So everybody in the hospital dropped what they were doing and started to do new things directed towards COVID-19. And so people who had that ability here worked on that. I think it also speaks to a little bit of a lack of coordination of how this would be attacked from the beginning. And so from the beginning, there was not a sort of, at least not a publicly announced kind of strategy that was clear to everybody in terms of how things were going to happen, how things were going to get laid out from sort of basic science, understanding some of those basic facts to driving it into clinical trials. And that creates a fair amount of duplication. And an example of that, just to answer the question you brought up, there are clinical networks that have existed, and there are lots of them that still exist. It's just that it took several months to utilize those for COVID-19. They sat not being used for COVID-19 trials for a period of time, and now they're starting to get used. And so it may not be creating a new network. It just means understanding that these things exist, and you have a strategy ready to go that you're going to use them.
Great. Thank you. Thank you. Dr. Stevens is fascinated by the notion that the COVID, the RNA translates into 30 specific proteins. You understand the structure, how they're folded, that 10 to 20 of them are what creates this issue. And you talk about purpose built. Are you thinking that is it possible to think, and I think mathematically with your supercomputers, about how to tear apart one of those 20 proteins?
Well, we're building both physics-based models and AI models to essentially custom design inhibitor molecules for each of those proteins using the power of supercomputing. I mean, that's what the community that can do this type of biophysical modeling and AI is working on. There's many groups that are collaborating on this task. And I believe in the near future, I can't say exactly how long this will take, we will have new compounds that are the result of this process that will go into the experimental screen pipelines.
Would you have to disrupt more than one of those 20 proteins to make
Yes, you might have to. If you look at state-of-the-art antiviral therapies, often they're a cocktail of drugs. And so we think the best strategy is probably a multiple therapeutic mix that would go after multiple targets, maybe a target that would help in blocking viral entry, one that might block replication, one that might block some host process that is a problem, and so forth. And so you would probably end up, at the end of the day, with a mixture of compounds in a future drug or future drug treatment. I think it's really important, though, to say that to develop the kind of drugs that we're imagining will take a long time. If you think about in the HIV timeframe, it took many, many years before there were effective HIV therapies, over a decade. While we're moving faster, we have better tools. This is a very hard problem. And while the community has been working on kind of crisis mode in this, they can't work in crisis mode for years. And so we have to put institutional structures around this to get it done.
Thank you very much. I yield back, Mr. Chairman.
Thank you. I'll recognize Representative Wexton for 5 minutes. Mute. Whoops. Microphone. Mute. You're muted.
Shame on me. Hi, thank you, Mr. Chairman, for yielding and for not charging me my 5 minutes quite yet. And to the panelists for joining us today. This is a really fascinating discussion, and I'm glad to be here for it. The controversy surrounding the FDA's EUA for hydroxychloroquine has caused a lot of people to question the scientific integrity of FDA's process. And so the policymaking, especially during this pandemic. Dr. Lurie and Dr. Rome, could you please give us your general assessment of the rigor of FDA policymaking and public communications regarding the repurposing of therapeutic drugs during this pandemic and what needs to be done better, if anything?
Well, I certainly agree that it has been a disappointment. And I don't think it's because of the career officials at FDA, who I believe are completely committed to scientific integrity and proper regulatory procedures in this pandemic. But I do think that people have turned out to be susceptible to political pressure. The hydroxychloroquine example is frankly an embarrassment. The standard for approving the EUA was considerably below what it was in previous EUAs, and I say this based on talking to people who granted EUAs in previous administrations. And so in the end, that turned out to be a black eye for the industry, for the agency. Another embarrassment, I think, has turned out to be antibody tests, where for a while, the agency allowed these products to come on the market without even an EUA. And then that turned out to be a disaster when it turned out that they were plagued by false positives. So now they have an EUA. What I hope is that from the combination of those two experiences, we'll get a proper use of the EUA process, which I think we're now seeing for remdesivir. And I sincerely believe and hope that my former colleagues at FDA will be able to stand up from the political pressure, because it is certainly searing.
Dr. Rome, how about you?
Yeah, thank you for the question. I think what you're getting at is actually an issue that has come up again and again, which is sort of the standards of evidence sort of required for any drug approval, even before COVID-19, which traditionally were statutorily supposed to be based on sort of substantial evidence. So it means that traditionally that meant 2 clinical trials, 2 large randomized clinical trials, to make sure that even if one trial got the answer wrong, that we wouldn't get it wrong twice. And that has changed over time. There are many drugs now that are expedited through the FDA process. And that's something that needs to be considered. And that's sort of the background for when COVID-19 came, we've already experienced the fact that 80+% of drugs are approved through some expedited pathway, maybe based on more limited evidence than that traditional 2 clinical trials. So during COVID-19, we've relied on the EUA to sort of cut off the FDA approval process and act before the FDA sort of carefully considers all the evidence. Makes total sense, and certainly time is of the essence. But to your point, we actually have very little experience using EUAs for drugs. It was done during the swine flu pandemic of 2009, 2010. There, a drug was issued an EUA, and the data that later came out from the clinical trials showed that the drug was not actually effective for the type of patients that EUA was issued for. Now we have hydroxychloroquine and remdesivir. So we've maybe hit 1 out of 3 potentially, though we don't know the full story on remdesivir. But certainly this is the time for Congress to take a look at the way the EUAs have been utilized and tune up the regulations on the FDA to make sure it's done appropriately in the context of the speed that's needed.
So in this final minute, I guess my question is also there's going to be a lot of financial incentive for various firms to get their drugs through this EUA process and get them approved. How does FDA assess and eliminate conflicts of interest in its drug approval process? Make sure that there's no conflicts in the decision-making process.
Well, quite frankly, the process is infected with conflict of interest, and there's not very much that can be done about it in the following sense. We accept the idea that drug trials of diagnostics or vaccines are conducted in general by the manufacturers themselves. And so it's a given that there'll be that kind of conflict of interest and that the companies will come in with an interest in depicting the data in the way that best suits them. But that's where the FDA comes in. That's where the FDA's review of the actual data itself, which no other country in the world claims to do, that's where that kind of review, that kind of insulating of the reviewers from the manufacturer is so important. And that's the way it's managed. But it's a given that most of the time the studies will have been done by the manufacturers themselves.
Thank you very much. I see my time has expired and I will yield back.
Well, thank you. And I will now recognize Representative Perlmutter to unmute and speak your side.
Thanks, Mr. Chair, and to the panelists. Thank you all for your testimony. I've been watching you all listen and watch each other, and it's been interesting. So the first question I have, and Dr. Finnegan, you brought up a couple networks, and you say there are been networks in place, but they really didn't get activated promptly. So you talked about active and you talked about PEDAL, I guess, sort of the acronyms for these things. And to everybody, but start with you, Dr. Finnegan. A pandemic comes, we see this thing starting to roll. How is there some lead agency? Is it CDC? Is it NIH? Is it BARDA? Who is it that says to these networks, okay, everybody's got to jump to, whether it's the hospitals or the laboratories, or who says, get going?
So I think to a certain degree, the exact agency that does it doesn't matter. I think it just needs to be understood ahead of time. And so whether or not it is the NIH or BARDA, or another agency, I don't think really matters. It's just a function of thinking ahead of time and knowing ahead of time that these networks exist and that you want to put them into action. And especially in a situation like this where you actually have some lead time. So we knew about this pandemic for some time and we could have been planning it. And so it doesn't necessarily matter that it's the CDC or a different agency. Whichever agency, let's say, funds that network, or if you need to bring in more than one network and you might need to have cross-agency, those things just need to be thought of ahead of time. Other aspects like how you would do consenting for patients and those things also should be thought of ahead of time so that can be streamlined as quickly as possible for when you need to utilize those networks.
So, Dr. Stevens, when were the labs sort of kicked into gear on this thing?
The labs started to self-organize around the 1st of March, ahead of the official proclamations. We have a little bit of internal flexibility at the lab. That's why the labs exist, to do large-scale science, interdisciplinary, and we're used to taking initiatives on our own, taking our own initiatives. So the labs started talking to each other. We started arranging computer time. We started pulling together teams, actually long before the pandemic was declared. And DOE headquarters was very supportive that we were already moving. And so I think the community, I think the others panelists would agree, the community saw this coming and started to do things that they could do within the realm of their degrees of freedom of action. And the agencies then came up to speed to start resourcing things. So I think, it all started happening in parallel.
So to all of you, from our science committee, we have a little bit of jurisdiction with respect to hospitals, but not a lot. But we do have definitely jurisdiction over the laboratories. So is there any connection in these networks that you talked about, Dr. Finnegan, or you talked about the 9 labs that you're collaborating with, Dr. Stevens? Is there any connection between sort of these hospital networks and our laboratories? Are you guys talking to each other?
Not so much. The Department of Energy typically doesn't get involved in clinical research. Our laboratory doesn't have ongoing internal projects related to clinical work. This is largely due to the distinctions between the different agencies that fund that work. We do have a lot of collaborators in universities. And so the way in which we, and I personally have a joint appointment at the University of Chicago where I have colleagues in clinical trials, and I have funding from my involves talking to those people. So personally, we have contacts, but institutionally, the Department of Energy, typically the laboratories that they support, aren't involved in clinical work.
All right, well, let me turn to Dr. Rome for my last 45 seconds. So at Harvard, so National Jewish collaborates with lots of hospitals, the University of Colorado, a bunch of other stuff. Harvard obviously collaborates with everything around the world. When did your medical school and when did you sort of get engaged in this thing? I mean, the minute we heard about it from China, or how did that go?
I agree with what's been said that the scientific community acted early, but acting early involves having information, and I think the information comes from the top in this case. The information out of China was challenging, I think, for medical professionals to understand. And so by the time in March that people started to gear up here, that might have been, unfortunately, pushed earlier had the administration and everyone else in the government sort of set the ball moving and pushed for action earlier on.
And I was remiss to say, Dr. Finnegan helped me on a telephone town hall where we had 10,000 people on the line. And so I just wanted to mention that and thank all of you for your testimony today. Really appreciate it.
Well, thank you all. At this point, I guess there's probably enough member interest for another round of questioning for those members who wish. And so we can quickly, all right, Ed, Dr. Perlmutter and Beyer. Okay, so this may be a brief round of questions here, but I would like to follow up on a couple of points. Dr. Finnegan, you sort of mentioned the idea, the concept that there, instead of having to replicate the approval of the remdesivir trial at 60 different locations, that there could be some single point of approval. And is it realistic to expect that individual institutions will buy into that mechanism, that they'll be willing to outsource the approval of a clinical trial that may or may not, be safe, which is one of their concerns?
So the answer is no, they're not going to give it up, but I think there's things you can do ahead of time to make it go much faster. So what happens with a trial like the remdesivir trial or other trials that we have going on is an industry sponsor in these instances reaches out to us, asks us if we want to be a part of it. We say yes. And then at that point, you begin the back and forth. If we get the protocol, we read it, it goes through our process. We have to approve it and make sure it's safe. We have to discuss the budget. We need to make sure we have to do it. While they're doing that with us, they're doing that individually at all the different sites. If we spend, let's say, 3 weeks working on it and then decide we can't do it, that's wasted time for everybody. So if you have a network like, let's say, the PEDAL network that I mentioned, this exists already in place. A lot of that work has been taken care of already. So a lot of that immediate work and legwork in terms of getting things approved and contracting and budgeting, that stuff might be taken care of. And so you can imagine a situation where a drug company might say, I have a drug, I think it's promising, I want to use this federally funded network, and maybe there's some mechanism by which they have to pay in to use that to help keep that funded, but allows them to very rapidly get their drug out there and not have to go through the process every time.
All right. And so, yeah, you touched on the issue of getting the commercial incentives right, because that must be very delicate in this, because, obviously, if a drug company can get the federal taxpayer to pay for a clinical trial for a drug that they'll eventually make money on, say this is something which is a currently unapproved use, you'd think that there ought to be a mechanism in place somehow to have the federal taxpayer, have some benefit from the fact that they paid for this trial. And are there countries anywhere that have a different model that might be more effective for dealing with the commercial interest to fund trials, or do they just fully federalize it and there's a big pot of money that and a group of scientists who decide what is the most scientifically promising and allocate some fraction of the clinical trials that way?
So I can't speak to what happens very knowledgeably in other countries. I'll let other people address that if they know that. What I will say briefly is that there are some examples of I don't know if it's quite public-private partnerships, but for example, in cystic fibrosis, the Cystic Fibrosis Foundation, which really sort of regulates a lot of the trials that happen in cystic fibrosis in kids and adults, partnered with an industry sponsor on new drugs so that they could rapidly get those drugs sort of tested, and then those were successful and approved. And so the drug company and the Cystic Fibrosis Foundation both benefited from that. And I think that's an example that could be replicated in other instances. I'll let others talk about other countries.
Dr. Brigham, any comments?
Yeah, I guess I would say that I don't know of in other countries, sort of to your question of sort of how that balance is made, other than other countries do a much better job negotiating for value-based prices of drugs, so that we do not sort of double pay, paying sort of through the roof for clinical research to develop the drug, and then secondarily for high prices. So that's one comment. The other is that not just in COVID-19, again, I mentioned the example of remdesivir, which is exactly what you said. A government is funding the late stage sort of clinical trial development. That occurs in one in four drugs that have been developed over the last decade, where the federal government is involved late in the development of the drug. And almost every drug has some sort of federal involvement in the early stages of development. So this is absolutely a problem. It's going to be highlighted in COVID-19, and it's going to affect how the drugs are able to be accessed by patients once we have to, once patients and insurance companies have to pay for them. So it's definitely something that needs to be addressed.
Well, thank you. And I think one of the most potentially tragic outcomes is that promising drugs just won't get looked into. That should, if you don't set this up right. All right. And at this point, I'm happy to recognize Chairman Lucas for 5 minutes of additional questions.
Mr. Chairman, I will yield back, but simply note our constituents out in the countryside are frantically looking for ways to protect themselves from this or cures to address it. I'd like to think that all the discussion we had today about the challenges we faced to this point, still the underlying issue is we're making progress, we're diligently working to address the needs of our constituents, and that they should have faith in the institutions, both inside and outside the federal government, that are working together, healthcare industry included, to try and address their needs that are brought on through issues of by no fault of their own, just the world we live in at this time. And with that, Mr. Chairman, I yield back.
Thank you. And I'll now recognize Representative Byer for 5 minutes.
Thank you, Mr. Chairman, very much. Dr. Stevens, in your testimony, you talked about us paying attention in the long term to emerging pathogens. How do you define emerging pathogens? Does someone have to get sick before we do it? And given the billions of different viruses that are out there, what constitutes an emerging one?
Well, there's several definitions, but the one that I think most people would agree with, it's pathogens that are relatively new to science. So these could be pathogens, zoonotic pathogens that are endemic in wild animals, maybe in places people don't normally live. And as humans, human development, economic development pushes large numbers of people closer to these previously wild areas, there's lots of opportunity for contact with these animal species, and new pathogens can emerge that way. That's the primary mechanism. If you think of over the last couple of decades, the viral pathogens, Ebola, Zika, SARS, SARS-CoV-2, these have all emerged from animal reservoirs. And so my recommendation is that we mount a scientifically based international program to surveil wild populations, understand their microbiomes, the natural viruses that coexist with them, and study them. We have the technology for doing that. We can easily sequence these things. We can produce the structures. We can get ahead of the curve. We can understand the reservoirs much better than we do currently, and that would leave us more time to be ready for the next one.
Thank you. You also mentioned that there are 10 million molecules available for experimental validation. You can't. And you talked about the winnowing, the funnel. Is the first funnel going to be a mathematical one, physical one, rather than experimental?
The top of the funnel, of course, in some theoretical sense, is every molecule that's drug-like. It's about 10 to the 60th. The drug companies and the academic community have an understanding of maybe 4 to 5 billion molecules. Of that, maybe 10 million are something I can get my hands on in a couple of weeks. Of course, synthetic chemists can make new things at roughly that time frame as well. So it would be conceivable to create ahead of time panels, that is, collections of molecules that are essentially frozen in the freezers. And if you have a an emerging outbreak and you develop assays quickly, you could screen a very large set of molecules in a few months and have a lot of possible leads to chase down. And combining that with computation would create a much better situation in terms of future therapeutic development. And that's something I think the community would be very excited to work towards.
Excellent. Thank you. It is amazing to try to get your arms around the 100 million molecules. One last question, Dr. Rome, your fourth point was making improvements to the FDA's EUA process. And you talked about the bullets, clarifying and standardizing, transparency, equitable distribution, and then patient outcome data. Is this something that should be done regulatorily, or is this a perfect piece of legislation for the House Science Committee?
So the FDA has the ability to do some of these things, but has not done so. They have the ability to collect information about the drugs, but are not required to, and other than adverse event or safety reporting of sort of major events like deaths that occur from the drug. So that has happened, but further data has not been required as part of the EUAs. The FDA has broad authority to write into the EUA what it wants in terms of requirements. But certainly those requirements, that sort of broad authority could be better regulated by Congress by sort of more directing and saying, when you issue an EUA, these are the things that we think are necessary. And we've learned a great deal about what would be helpful. I would say, again, more transparency at the outset so that physicians who are using the drugs from an EUA have access to data, not just the data that's, on the internet, but actually like the raw data and the published studies really help.
And you wouldn't think that we were guilty of micromanagement if we led that?
I think that these things need to happen, and so I think if Congress wants to step up and say that for all future EUAs, that these are some tweaks that we think are necessary, I think that's reasonable. I would say the EUA applies not just to drugs. The EUA applies to diagnostics, testing equipment that has been mentioned before, ventilators, other things. So again, drugs are a minority of cases over the history of the EUA that have been where it's been used. So we only have really 3 examples, 2 of which for this pandemic alone. So we are I do think it's time to reevaluate how that was used and decide if small changes, legislative changes need to be made.
All right, thank you very much. Mr. Chairman, I yield back.
Thank you. And I'll now recognize Representative Perlmutter for 5 minutes.
Just one question, and start with you, Dr. Lurie. Anybody can jump in. The serologic tests, the antibody tests, I guess. There are a whole bunch of them. Some have been approved, I guess, and some have not been approved. So going to the diagnostics that you were talking about, Dr. Rome, did we do this right or not? Or is there now sort of doubt about these tests and their validity? Dr. Lurie.
Well, in referring to the antibody test specifically, I don't think that has been well handled by FDA. I think that feeling the pressure to press ahead, they gave the antibody tests a free pass to begin with, and not even requiring an EUA for them. And it didn't take long for evidence to mount that, particularly in low prevalence populations, certain of these tests could actually produce more false positives than true positives. And I think that forced the agency to take a look at what they had done, and then they slapped an EUA upon them, giving the companies 10 days to comply. I don't know how many of them have met the EUA requirements at this point, but I expect that certain of those products just disappeared because they couldn't meet the standards. So again, what I'm sorry to say has happened is that the FDA has been kind of flip-flopping, or if you like, course-correcting, to try and get this exactly right. They've made some mistakes. Hopefully, it will be better going forward.
Okay, thank you. I don't know if anybody else has any comments. I just want to thank the panel, and I'm happy to yield back to you, Mr. Chairman.
Oh, thank you. And I have to say that I was a little bit surprised when the antibody tests came out so flawed, that there was no one responsible in the government for establishing a test panel that you run every one of the proposed tests against, just blood samples of positive and negative people that would you be just prepared and at least given to every manufacturer to test against and report their results. And that seems like the sort of infrastructure that should exist somewhere in the future when this sort of thing happens. And I was also fascinated by Representative Byers' suggestion of potential legislation coming out of this, that there may be something sensible to be done there. And let's see, I can have about one remaining minute of Representative Perlmutter's time. So I'd just like, Dr. Stevens, how leaky is the funnel? How often do you see a drug that works wonderfully in practice and not at all in theory?
Well, usually it means our theory is wrong, so we have to go back and fix the theory if that happens. The funnel's pretty leaky in the sense that things fall out that we have to filter out because we're using approximate rules often to do this. But most of what we look at doesn't work. I mean, that's the reality of drug development. Most compounds don't work. And so it is a needle in a haystack type of problem. And occasionally you'll find drugs that defy our initial view. But those usually don't come from the computational process because we filter those out. They would come through physical screens on natural products, for example, and then we have to go back and rethink them. So you need combinations of both. This can't all be computationally driven. As I mentioned before, large chemical libraries that would be screened in public, I think, is another resource that we need as infrastructure.
All right. Well, anyway, I just want to thank all of our witnesses at this point before bringing the hearing to a close. It's very important, and keep thinking as you're doing your daytime job here, what changes you'd like to see in place for the next pandemic, because I think that's going to be a big part of our job, is to try to preserve the attention span of Congress so that we're better prepared. I guess someone smart once said, "You go to war with the army you have," and next pandemic, I'd like a slightly better army. And just thank you all for being part of the Army that we have. So the record here will remain open for 2 weeks for additional statements from members for any additional questions that the committee may ask the witnesses. And the witnesses are now excused and the hearing is adjourned.